My name is Sarabia. For this video, I'm going to briefly explain the DNA replication of prokaryotic cells. Scientists first used prokaryotic cells to explain the DNA replication, and later, they studied the DNA replication of eukaryotic cells. DNA replication begins in the origin of replication. This site, which has active replication called replication fork. DNA helicase, which is an enzyme, this one, separates the two single strands by breaking the hydrogen bonds between the bases, and single-stranded binding proteins stabilize the newly single-stranded regions. This initiates the DNA replication. DNA gyrase makes sure that the double-stranded outside the replication fork will not supercoil. Once the replication fork is stable, DNA polymerase, which is an enzyme, will catalyze the addition of new nucleotides to the growing strand. Beta clamp and Y complex holds the DNA polymerase to make DNA in place. RNA primers are paired to the template strand by an enzyme called primase, which will serve as a platform for DNA polymerase to extend and synthesize a complementary strand. Replication occurs at the same time in both strands. One is continuous and the other is discontinuous. Continuous synthesis is called leading strand synthesis, occurring in 3' to 5' oriented parent strand or the leading strand. It synthesizes a new complementary strand in the 5' to 3' direction. This continuous synthesis is the lagging strand synthesis, where DNA polymerase synthesizes new complementary strand in a back stitching because the parent strand is oriented 5' to 3', which is the lagging strand. The synthesis is done through the segments called Okazaki fragments. Replication on this strand makes use of primers added by primase ahead of the 5' and lagging strand, which occurs in a slow rate. DNA polymerase 1 removes the RNA primers and replaces it with the deoxyribose nucleotide. And ligase identifies and seals off the nicks and gaps present between the adjacent Okazaki fragments. To clearly show you the process, please watch this animation. A terminator is encountered by the leading and lagging strand to stop the replication. The terminator is a special sequence of nucleotide that signals the end of replication. The special protein blocks the helicase when it binds to the terminator, causing the helicase and other binding proteins to fall off. The DNA replication results to two identical copies of DNA molecule which are semi-conservative. That's all for this video on the DNA replication of prokaryotic cells. This has been Adessa Sarabia leaving you a message that with faith, perseverance to follow the process, and hard work, anything is achievable. Thank you for watching and see you in the next video.